Hello. We're going to continue talking about the citric acid cycle. The citric acid cycle is a series of 8 reactions, most of which are amphibolic. It is also known as the Krebs cycle, or TCA, or tricarboxylic acid cycle. Many of its intermediates are used in anabolic pathways as building blocks, and many molecules can enter this pathway throughout different locations. The goal of the cycle is to both oxidize acetyl-CoA and to regenerate oxaloacetate, so that the cycle may continue. 2 carbons enter as an acetate molecule, and 2 carbons are released as carbon dioxide, which we saw in the previous video. So we've already talked about the first 4 steps, which oxidize the acetyl-CoA molecule, so we'll now focus on the regeneration of oxaloacetate from succinyl-CoA. Succinyl-CoA synthetase converts succinyl-CoA into succinate. This is a very favorable reaction. It's actually named for the reverse reaction, hence the name synthetase, due to the hydrolysis of NTP in the reverse direction. This molecule also uses coenzyme A as a leaving group to drive the reaction. The succinyl-CoA becomes succinyl phosphate, which coordinates the phosphate's attachment to the enzyme histidine 246. The enzyme is then able to catalyze the formation of either ATP or GTP. Succinate is formed first before the nucleotide triphosphate. This reaction forms either ATP or GTP, which is dependent on the cell type. So tissues like the brain and heart form ATP in this step, while liver tissues form GTP. Both ATP and GTP are energetically equivalent, however, they're used for different processes depending on enzyme specificities. The 6th step is catalyzed by succinate dehydrogenase complex. Succinate is oxidized to fumarate, and FAD is reduced to FADH2. Fumarate has a trans double bond. This is very important. This is complex 2 of the electron transport chain, and is the location of FADH2 entry into the electron transport chain. The FAD prosthetic group is covalently linked to the succinate dehydrogenase complex and gives the electrons that are extracted from succinate to the iron-sulfur complexes of the electron transport chain. The electrons will travel a very long distance of around 40 angstroms before the enzyme reduces ubiquinone. The FAD molecule is essential to this reaction. Unlike other reactions which utilize NAD+ as the electron carrier, this complex uses FAD. Therefore, succinate dehydrogenase is classified as a flavoprotein. The 2 electron carriers seen here are very similar in structure. They appear as modified ADP molecules. They have adenine, cyclic ribose, and 2 phosphate groups. The difference is that attached to the beta phosphate is either a cyclic ribose, which is attached to niacin, or a linear ribose, which is attached to flavin. Both niacin and flavin are essential vitamins. FADH2 is actually lower in energy than NADH. That's why this allows the carbon-carbon bond to be oxidized in succinate dehydrogenase. Less energy is required to reduce FAD to FADH2, therefore the reaction can proceed. If this enzyme was dependent on NAD+ to be reduced into NADH, the reaction would require the input of energy to proceed. For NADH molecules created in the cell, oxidative phosphorylation via the electron transport chain will produce about 2.5 ATP per molecule, while FADH2 produces around 1.5 ATP per molecule of FADH2. The 7th step is catalyzed by fumarase. This reaction is reversible. The reaction is semi-favorable, but the energy is not directly captured here. Fumarate is hydrated to create yet another hydroxyl group that can be oxidized to produce even more ATP later. As you can see from the mechanism on the bottom, that there is a carbon ion formed as an intermediate. L-malate is the product, which is the exception to the typical conformation of carbohydrates, which usually use D isoforms of sugars. This enzyme is extremely stereospecific. Malate, which is similar to fumarate with a cis double bond, will not react, and D-malate does not react. The 8th step and final step is catalyzed by malate dehydrogenase. This reaction is highly unfavorable. This is one of the reasons why oxaloacetate is extremely low concentrations in cells. The equilibrium of this reaction is pulled to the right due to the low concentration of oxaloacetate. NAD+ is reduced to NADH, and electrons come from the hydroxyl group, which was just added in the previous step. This reaction is really important since it regenerates oxaloacetate at the starting point of this cycle. So now that the cycle has regenerated oxaloacetate, it's ready to begin the cycle again. Some things to remember about the citric acid cycle is that 3 NADH are generated, which make around 9 ATP. 1 FADH2 is generated, which makes around 2 ATP. 1 ATP is made directly via substrate level phosphorylation. Many of these steps are reversible, and the reactions rely heavily on good leaving groups such as coenzyme A and carbon dioxide to proceed through the cycle. Thank you.